Welcome to the fourth webinar on topics associated with human islet isolation. Today's topic is an overview of the pancreatic extracellular matrix. I'm pleased that we could get Dan Tremmel, who's a PhD student just finishing up his degree in John Odorico's lab at the University of Wisconsin. What Dan will do is review the current state of the art, but more importantly, how these new proteomic tools are complementing our understanding of the extracellular matrix. My plan is to continue having these webinars on a quarterly basis. If you have any suggestions for a topic to discuss in these webinars, please do not hesitate to contact me. If you are now looking at older webinars and have questions, also contact me and I'll get in touch with the earlier speakers about answering your questions. Just housekeeping, ask at any time you can ask questions using the chat function during the webinar. We will discuss and cover all of the Q&As at the end of the webinar. This webinar will take about 35 minutes. There will be a poll question at the beginning of the webinar and 2 poll questions during the Q&A session of the webinar. With that, I'm going to sign off and hand it off to Dan to start the presentation.
All right, thanks, Bob. I am going to turn off my video and share my screen. Okay. All right, thanks for the introduction, Bob. As he said, I'm going to talk about an overview of the pancreatic extracellular matrix. And I'm a graduate student in John Odorico's lab at the University of Wisconsin. And I know that everybody here is familiar with islet isolation. So just some very brief background about what I think everybody already knows. In order to recover islets from the pancreas, collagenases and proteases are used to digest the ECM of the organ and free the islets. A recent study has found that greater than 60% of the collagen needs to be digested in order to increase the odds of having a successful islet yield and being able to be transplanted. And we know that during this process, the basement membrane of the islets is substantially disrupted in order to free the islets from the tissue. So a little bit of an introduction to ECM in general and why we're interested in this. ECM is a network of proteins and polysaccharides that fills the extracellular space around the cells within tissues, and it can interact with cells in a variety of different ways, including directly binding to membrane receptors such as integrins and stimulating downstream signaling. But the ECM can also bind and regulate growth factors throughout the extracellular space and influence their signaling to cells. And I think most directly, the ECM provides structural and mechanical support for the cells. And interestingly, ECM composition is unique within each type of tissue. So when it comes to understanding how the ECM is supporting cells, or in the case of islet isolation, being able to digest the ECM to free the cells, that is a unique material that's going to be different based on the tissue and based on the species that you're working with. So in my conversations with Bob leading up to this talk, he used the analogy that the matrisome is a jungle, and I like that analogy a lot. I think for a couple different reasons it's appropriate. First of all, the ECM is made up of hundreds of different, or could potentially be made up of hundreds of different proteins that fall into the category of ECM. So it's a really complicated material, and on top of that, there are polysaccharides and different protein modifications that can be added to those proteins that affect the structure and the signaling of the proteins themselves. But I also think that the 3D structure of the proteins is also sort of similar to a jungle in a way. And you have this dense network of different protein chains and very, very large fibers that are formed that can be made up of different ECM proteins. And so this is really sort of an uncharted territory where there's a lot more that can be understood. And so that's what I'm going to talk about today, mainly with respect to understanding the protein composition of the ECM in the pancreas. And this work really started a long time ago using some different methodology. And I think some recent studies have employed broader methods that can characterize the ECM a little bit better. But also a lot of the older studies were using different model organisms. And I think it's become pretty clear over the years that human systems and definitely human islets are different than some of these model organisms. And so when we're talking about the ECM, we want to know what the differences are, and more specifically, what the ECM of the human pancreas and human islets is, because better understanding this complex mixture of proteins is going to be able to help us to improve islet isolation technology. And in the work that we do in the Odorico lab, we're really interested in how we can better understand ECM to support islet health and function. So as I said, the ECM is damaged in the islets during the isolation process. And we have found, and many others have found, that if you can replenish the ECM in those islets after isolation, you can improve their survival and function in culture and after transplantation. And then I also think it's important to consider the role that ECM may play and changes to the ECM that may occur in different disease states. So a disease like pancreatitis may, because there's fibrosis in the pancreas, may have a more direct role for ECM, but there have been, there are changes in ECM associated with different types of diabetes as well. And it's important to understand those both in the context of the disease, but also in order to improve islet isolation from donors with those conditions. We're also interested in looking at how the ECM changes with development. And in our work, we've looked at very early stages of development, but for the context of this presentation, I think it's important to consider that potential organ donors still represent a pretty large range of ages. And so understanding how the ECM may change from younger to older adults may also be able to benefit islet isolation. And what I'm going to talk about in this presentation is, first, I'm going to go over some methodology that has been used to characterize the matrisome. And then I'm going to talk about quantitative studies of human pancreas ECM protein composition. And then a couple studies that have used collagenase and protease digestion in order to better understand ECM in human pancreas. So one of the more common methods that has been used to study the ECM, especially in older studies, has been using immunostaining. And I think this is something that most people are pretty familiar with. But just to cover the basics, we can use antibodies for a specific antigen. So in this case, an ECM protein. And when the antibody binds to that antigen, the antibody can be labeled or detected in some capacity, and then you're able to visualize the localization of that protein within the tissue. So this is a pretty common method of looking at proteins, and it's really limited based on antibody availability and specificity, because if the antibody isn't specific enough to the antigen that you're interested in, you may get misleading results. But also a lot of times these antibodies are specific to only certain species, and so it's important to select the antibodies appropriately. And then with ECM proteins in particular, there's a high level of sequence homology between the proteins. So in order to detect these proteins, antibodies have to be generated specifically to the unique segments of the different collagen proteins, for example. And that requires, I think, really good characterization of the antibodies in order to be sure that we're looking at collagen 1 and not having any overlap with collagen 3, for example. And then, of course, like I said, if the antibody is binding to an off-target antigen, then you can also get misleading results with your staining. So A different approach at looking at protein composition is using mass spectrometry. And here proteins can be digested into peptides, which can be input into the mass spec, and these are ionized. And then at the end, the mass to charge ratio of the ions is detected. And this is a unique identifier for the different ions in the system. And so looking at the spectra and looking at the mass to charge ratio, it can be relatively quantified for the abundance of the different ions in the sample that was input into the mass spec. And using, I guess technology has really improved with mass spec, using a known protease that has a predictable cleavage site such as trypsin, combined with protein sequences and libraries of mass to charge ratios for known tryptic peptides. Really, really complex samples can now be used with mass spec. So, for example, in our work, we're using tissue lysates that are all of the proteins in that tissue combined. And you get obviously very, very complicated spectra out of that, but using really sophisticated technology, software, you can analyze this and do relative quantification for the different proteins that you're starting with. Another approach that has been used in the last couple of years to look at the ECM in the human pancreas, and particularly in the context of islet isolation, is Raman microspectroscopy, or RMS. And here, the sample that was used in these studies is a tissue section, and shining a laser of a known wavelength onto this sample and then detecting the scattered light that is reflected off of the different molecules in the sample, and measuring the shift of wavelength based on that detected light, you can predict the molecular bonds that are present in that sample, essentially. And then using known compounds and establishing a library of Raman shift for those known compounds, you can assign a chemical bond to the different wavelengths that are generated. So we'll talk about this a little bit more when we get to those studies. All right, and comparing these three methods, I think they are useful in very different ways for studying the ECM. As far as how high throughput they are, I think the methods that are using tissue sections are a little bit more limited because you have to have a slide and a section for each sample that you're looking at, where mass spec now has a lot more multiplexing options. And of course, all of these methods have variations that may be better suited depending on the outcome that you're looking at. I know at least with mass spec, there are a lot of different variations that you can use. And with protein specificity, immunostaining, as we discussed, depending on the antibodies used and how well characterized those antibodies are, they can be very reliable for detecting the protein that you're interested in. But without good characterization, there can be some uncertainty about the results from immunostaining alone. The RMS, I think, is using those Raman shift values to predict the types of proteins that are being studied, but is not necessarily ideal for detecting a specific protein or a specific type of ECM component, for example. Whereas mass spec can really be reliably used to identify a specific protein in a sample and to quantify that protein. For ease of use, I think immunostaining is probably the most commonly used and the most approachable method for most labs, although RMS and MS require pretty sophisticated equipment and especially software for analyzing. But those are becoming more commonly found in universities and companies. And so I think with modern technology, those are becoming a little bit more approachable for people. And then for tissue visualization, the IF and RMS are using tissue sections and generating images based on those. And so Those are good for visualizing the localization of some of these proteins within tissues. And traditional methods of mass spec aren't able to do this. For example, you may be just using a protein lysate, and so then you're just mixing together all of the proteins from the sample. But there are mass spec imaging techniques that are available now that do give you some localization information as well. Okay, so going back a few years to some earlier studies about the ECM of the pancreas, immunostaining and microscopy studies established some early knowledge about the ECM of the pancreas and how this differs between different species. Electron microscopy found that there were differences in the thickness of the ECM at the exterior surface of islets in different species, and using antibodies specific to certain ECM proteins, found that there were some differences in the ECM composition of the pancreas in different species. And among a lot of different studies, this is a very good review, the Stendhal et al. Paper from 2010. It was sort of generally identified that laminins, collagen 1, 3, 4, 5, 6, and fibronectin are fairly abundant in the pancreas and islets. And this data was derived from a lot of different species and a lot of different studies, and they didn't always agree on which ECM proteins were most abundant, for example. But at least a list of ECM proteins that were present in the pancreas were identified using these techniques. And after that was established, groups looked a little bit more closely at more species of proteins. So, for example, here are a couple of papers that looked at a lot of different isoforms of laminin and found that some of these laminin isoforms are found throughout the pancreas and in the islets. Some of them are only found in the vessels, for example, and not found in the islets at all. So this helped to narrow down what types of laminins were present in the pancreas. And comparing the mouse to the human, they also identified, really interestingly, that the human pancreas has a double basement membrane around the blood vessels through the islets that was not found in the mouse pancreas. And so a schematic like this sort of represents some of the differences that were found in the basement membrane and the protein compositions of these between mouse and human. And this is definitely beneficial to studies that are interested in the ECM of the islets and particularly of the human islets. But I think immunostaining studies are a little bit limited in fully defining the composition of the human pancreas matrisome because they're selecting specific antibodies that they want to look at, and they're biasing their research just based on the antibodies available and the antibodies that they want to use and the proteins they want to look at. So this sort of left open some broader studies that want to define the composition of the human pancreas, both by identifying the total composition of ECM proteins present and also by relative quantification of which of these are most abundant in the pancreas and in the islets themselves. And as I get into some of the mass spec data, I wanted to outline some of the categories of proteins that are generally used when looking at these big data sets. Projects such as the Matrisome Project and MatrixDB database have established groupings of these different matrisome and matrisome associated proteins based on their localization and function in tissues. The NABA group in particular has done extensive work to look at all of the different ECM and ECM associated proteins in the human and mouse proteome. So the core matrisome genes, which are split up into glycoproteins, collagens, and proteoglycans, these are the more structural ECM proteins. And so you can see you've got laminins and fibronectin and collagens, and then proteoglycans such as osteoglycin, biglycan, lumican, et cetera. The matrisome-associated genes are proteins that are going to be found in the extracellular space, but not necessarily a part of the ECM. So that includes secreted factors, which are going to be like growth factors, chemokines, or insulin. ECM regulators are often proteases or MMPs or inhibitors of proteases. So these may interact directly with the ECM, but they're not structural parts of the ECM. And ECM-affiliated proteins are proteins that may directly interact with the ECM, but again, aren't structurally part of the ECM. So that also includes some ECM receptors. Okay, so hopefully that will help to sort out what we're looking at, but really I'm going to focus more on the core matrisome because this is the structural part of the matrisome, and that's probably what's most relevant when it comes to islet isolation. So I'm first going to talk about the biomaterials study that came out this year from Giuseppe Orlando's group, and they used 15 human pancreas donors aged 16 to 58. This study used decellularization of the pancreas as one of their tools for enriching the matrisome, or enriching the ECM component of the tissue. But I didn't want to include too much of that because it's a lot of pie charts and a lot to look at. So I wanted to focus more on the data that they generated that's related to the pancreas matrisome, and not so much looking at this decelled material that they've generated. So the first chart here is the whole pancreas tissue that they looked at through mass spec. So you can see that they identified in gray a majority of the tissue with cellular proteins, which makes sense. They did find that by protein abundance, the core matrisome made up 22% of the pancreas, which is a pretty large number. And if you look over here, I've got some animations here. That the core matrisome includes the glycoproteins, proteoglycans, and collagens. It's mostly collagen that makes up the core matrisome by abundance. By the number of proteins associated with these different groups, they identified 37 structural ECM proteins in the human pancreas samples. And that was composed of 16 glycoproteins, 7 proteoglycans, and 14 collagens. But again, by abundance, the collagens are the majority of this group, and the glycoproteins and proteoglycans are a really small fraction, but there are a lot of different species of proteins detected. So as I said, they used a lot of different methods in this paper. They used their decellularized pancreas, and in some ways that enriches the ECM fraction, and so it enables detection of some lower abundant species of ECM that you might not get in the whole pancreas lysate. They also used some ELISA data, and when they combined all of their data together, this was sort of their final result. So they identified 314 matrisome proteins, and 71 of those are structural. 16 collagens, 39 ECM glycoproteins, and 16 proteoglycans. And out of the entire proteome, by number of proteins, the structural ECM made up 5% of the proteome. And I think this is very useful information in some ways, but in other ways, the way that this is presented in this paper, it's a little bit difficult to look at the abundance of individual proteins. So they did collect quantified abundance data for the different groups, and they also identified a lot of ECM proteins. But if you wanted to compare, for example, what fraction of this was collagen 1, that's not really presented in their paper, but I'm sure that's possible to calculate using their raw data. So it's a little bit difficult to identify which ECM proteins are the major players in the pancreas. And also they had a pretty broad range of donors in this paper, but they didn't analyze these based on donor age. So another study that used mass spec and was published in the last year on human pancreas, and this is from our group, the Odorico lab in collaboration with Dr. Lingjun Lee's lab here at the University of Wisconsin. And we were interested mainly in identifying changes in the matrisome throughout development. And so we started with fetal tissue, and we used juvenile donors age 5 to 16 years old, young adult donors in their 20s, and older adult donors in their 50s. And the key aspects of this from a mass spec perspective utilized some techniques developed in the Lee lab. So we used a special preparation method to solubilize the ECM proteins better, to get better signal from the ECM fraction, which is generally highly insoluble, so sometimes it's difficult to detect. And we also used their DILU labeling method, where we use these different labeled DILU tags, and we can run up to 12 different samples at the same time. And this improves the relative quantification of the samples because they're going through the machine at the same time and they're loaded together. So that enables us to have relatively accurate quantification of the proteins between these different donors in the different age groups. All right, and a little bit about why we looked at these different age groups. Like I said, our lab is interested in the ECM for the health and the function of the islets, and also in the context of stem cell derived islets. So it's pretty well understood that the fetal tissue contains immature islets that lack response to high glucose. In the first year of life, the islets undergo functional maturation where they do become glucose responsive, but other studies have shown that up until about puberty, there's still an increase in glucose responsiveness and expression of certain maturation genes in the islets. So that's why we were interested in these age groups. And then other studies have looked at differences between younger and older islets, younger and older adults when it comes to islet isolation, and found that there are differences in the islet ECM of these different aged donors. So we were interested in what we could see in the differences of the ECM between these groups. And then, of course, we're interested in looking at how the ECM may differ between the acinar and the islet compartments of the pancreas as well. So to use some of those familiar pie charts, among all of the donors that we looked at, so that's the whole range of age groups, we did identify 19 collagens, 35 ECM glycoproteins, and 7 proteoglycans. So these numbers are actually pretty similar to that of the paper from the Orlando group. When we looked at this across all of the different age groups, so we actually identified 185 ECM proteins, and of those 117 were common to all of the age groups. So this is a part of our study where we're interested in how the ECM is changing from fetal to adult. And so we did lose some of those ECM proteins for quantification because they have to be present in all of the samples in order to be quantified. But of those 117, we found that 84 of them significantly changed throughout development. So this shows how dynamic the ECM really is. And of course, we're talking about fetal to juvenile to adult. And so those are pretty dramatic changes in the tissue. And we looked at the ECM proteins that were significantly different between those different pairs of age groups. And interestingly, the only one that did not have any significant differences was the older versus younger adult group. But we will look at that a little bit more in a couple slides. So then what I did was I identified 16 structural ECM proteins that seemed to be interesting, either based on their total abundance, having very high abundance in the pancreas, or based on their changes with development. And we did immunostaining to first validate the mass spec findings. And we found that most of these, all of these ECM proteins, the staining patterns and the intensity of the staining correlated with what we found with mass spec. So that was good. And then we used these images to further look at how the ECM changed between the islet and acinar compartments of the pancreas. And I actually really like these images too, because looking at laminin A4 and laminin A5, you can see how dramatically some of these components change with development. So laminin A4 is really abundant in the fetal pancreas, I think particularly in the developing blood vessels, and has decreased abundance with age. Laminin A5 has lower abundance in the fetal tissue and has pretty equal abundance in the postnatal tissues that we looked at. So we did find some very dramatic changes with ECM at that developmental scale. So now we'll get into the islet acinar aspect a little bit. So here on the left side, we're plotting the total intensity by mass spec in the black bars, and the purple bars are plotting the islet acinar ratio. So this will be above 1 if the protein is enriched in the islets compared to the acinar, and it'll be below 1 if the ECM protein is enriched in the acinar compared to the islet. And the heat maps on the right, I think, are a little bit of an easier way of visualizing this through the developmental changes. So, for example, certain collagens, collagen 1, 3, and 6, were highly abundant in the pancreas at all stages of development. So this is really interesting, especially for some tissue engineering work with stem cells. These are the common ECM proteins that are most abundant at all of the points of development that we looked at. A lot of other ECM proteins had pretty significant changes at the different developmental time points that we studied, but related to islet isolation, as I said before, there weren't any ECM proteins that had significant differences between the younger and older adults at the total lysate level. When we looked at the islet acinar ratio, we did find that there were some significant changes in the localization of these proteins throughout development. So, for example, some were more enriched in the islets in the fetal tissue, and some were more enriched in the islets in the adult tissue. But interestingly, there were a lot of significant differences in how these different ECM proteins localized in the islets and acinar between the younger and older adults. So this is what may be relevant to different islet isolation preps between these different age groups. So here I'm looking at this a little bit more closely. So as I said, there were no differences in the whole pancreas ECM content for any of these proteins, but the localization of collagen 1, 5, 12, laminin A4, and laminin A5, these were all more enriched in and around the islets in the older adults compared to the younger adult donors that we studied. And interestingly, collagen 6 was highly enriched in and around the islets in both the younger and older adult donors, with no significant difference between these two. So hopefully that stimulates a little bit of conversation or thought from the islet isolation people and the people who are looking at the different collagenase enzymes, because I know that there have been differences in islet isolation between these two age groups. And so it does look like the peri-islet ECM has a significantly different composition between these. So maybe we can talk about that a little bit more. And I think just a personal side note, I tried a few different fibronectin antibodies, and I am not really seeing much fibronectin in the islets or even most of the acinar tissue in the pancreas. We find the fibronectin is mostly at the interlobular interfaces and associated with large ducts and vessels. So that's just one of those things where I looked through some of the literature about ECM proteins, and I think you have to be a little bit careful when you're looking at the existing literature, because a lot of this data has been derived from other species. So I know that fibronectin is definitely present in the mouse islets, and sometimes that's referenced to apply to human islets as well. But at least for now, I have not really seen that the fibronectin is a major component of the ECM of the islets in the human pancreas. All right, so shifting gears a little bit to some papers that have used ECM digestion in order to better understand the ECM of the pancreas. This is an older paper that used this method of treating tissue sections with collagenases, neutral proteases, or combinations of these in order to see which ECM components are being targeted by these different enzyme blends. And in this paper, they used a Sirius red stain, which stains fibrillar collagens, and a periodic acid Schiff stain, which stains glycoproteins. And they could see that these were differentially digested with the different enzymes, but this isn't the most informative way of studying which specific collagens or glycoproteins are being affected, because these are broad histostains. So a more recent study by Paul Johnson's group, the Spears et al. 2018 paper, they used a similar method where they treated tissue sections with collagenase, neutral protease, or a combination. And they were looking at younger and older donors. So they used age groups above age 45 and below age 35. And they did find that when they looked at individual ECM proteins, there were differences in digestion with collagenase or neutral protease based on the donor age. And so they stained these before and after the different treatments. But when they combined the two enzymes together, this ameliorated the difference that they found based on donor age. And so there was even digestion of all of the donors. And they looked at this a little bit more in depth in their next study, which came out in 2019. And this is where they used RMS in combination with this digestion assay. And so here they used purified ECM proteins to establish this signature Raman shift profile. And they can identify some of these shifts, or they can assign some of these to specific molecular bonds and features of the ECM proteins themselves. And using this profile, they identified this ECM component of their tissue using RMS, and they were able to visualize this again without any enzyme treatment or with collagenase-neutral protease or the combination. And they used younger and older donors. I think the ages were slightly different here, 30 years in the younger group and above age 55 in the older group. And they found that when they analyzed the spectra of these using RMS, they clustered into two separate groups that were significantly different based on principal component analysis. And so I think they also measured only at the islet exocrine interface. So they're really looking more closely in this study at the ECM of the islets themselves. So these were differentially digested with respect to donor age. They also looked at the ECM, so the blue X's here, at the peri-islet interface compared to the intra-islet ECM, the red X's here, among different donors. And they found that the RMS spectra of these also were significantly different. And this was mainly related to peaks that they could assign to collagen signal. So not necessarily a specific collagen protein, but from a collagen-like spectra. So again, they're identifying that there are differences in the ECM based on donor age and based on the localization of the ECM in the tissue. And so one last paper that's also looking at these components in a similar way is the Meyer et al. 2020. And here they looked at the collagen content of a variety of different donors before and after they digested. And they also followed up by looking at the success of the islet isolation in those different donors. So they were wondering whether the collagen content of the pancreas affects the islet isolation and if that varies based on donor age or donor BMI. And what they found was that the collagen content did not vary based on donor age or BMI and didn't have a difference in digestibility. But they did find that based on the amount of the original collagen that was digested, so this is percentage of initial pancreas collagen digested, if they digested more than 60% of the initial collagen content, they had better islet isolation success. And this was based on the number of islets recovered and whether or not they were able to transplant those islets. When they digested less than 60% of the collagen, there was a high rate of failure of those pancreases. And so that's some useful information, but they didn't identify differences based on donor age on the digestion of the collagen. They did look at collagen 6 using immunostaining, and they did find that collagen 6 content was significantly different between the younger and older donors. But I do think they seem to have a relatively low N at the younger age here. So I'm not sure if a bigger group of donors would be necessary to make more conclusions about that. But I just thought that I would mention that. It's just a little bit different than some of the other studies. All right, and so to wrap this up, the ECM composition of the human pancreas has only recently been explored in depth. And what we found in our study is that the ECM changes dramatically throughout development, but in adulthood, no single individual ECM proteins were significantly different between younger and older adults based on total abundance. But based on the immunofluorescent staining studies and based on the enzyme digestion assays, It does seem that the ECM of the islet acinar interface may be significantly different between older and younger donors. And again, in our study, we found that collagen 6 was abundant in both, but collagen 1 and 5 were more enriched in the older adult donors. I do think that further studies are needed to look more closely at these localized differences in the ECM based on age and also based on disease state. It's probably interesting to look at what other donor variables might affect the islet ECM in order to better select donors for islet isolation or in order to better prepare enzyme blends based on donor variables. And just to sort of tie things back together, back to the matrisome is a jungle analogy that we made earlier. I think some of the next steps on this front are to look at how the ECM may cross-link differently or may have different post-translational modifications with age or in different disease states, and how this might affect the 3D structure, because when it comes to islet isolation, that's really what the enzymes need to disrupt, is the 3D structure of the ECM. And that's not only affected by the different proteins that are present, but it's also affected by the cross-linking and the modifications to those proteins. And on that note, I just want to quickly mention our collaborators in the Lee lab here, Dylan Tabang, just published a paper using our pancreas lysates, where he looked at post-translational modifications of the ECM proteins of the pancreas and found that many of them actually had phospho and glycosides, which isn't surprising because it's known that ECM proteins are post-translationally modified. But so he's really interested in better characterizing the PTMs of the ECM of the pancreas. And I think his next steps are to look at different ages and different disease states. And so this might be relevant to what I was just talking about. I'm going to go back to the summary slide, but I think I have to actually end my presentation in order to go back to the main screen. So I'll probably stop that. All right. Bob, you're still muted.
Thank you, Dean, for a very good presentation. What I would start out with is I can ask a couple of questions I had on my mind. I think it's relevant because we struggle as a manufacturer, of course. We're always trying to understand the science that you just described on how to improve enzymes. And I think what we get into is the whole quandary in biology is a holistic versus a reductionist approach. So we always like to find the pieces that work and decide what does this tell us about the bigger things. And it's as you've very nicely presented, in a very complicated organ structure or tissue structure, it's very difficult. And the way I look at it, and maybe if you're going to expand upon it, I think a lot of the reductionist approaches can easily be tested, as you indicated, by better islet viability, making the islets happy in culture or in some scaffold that you're going to transplant or whatever. But to me, it's still a quandary of how do we make better enzymes, because we need bigger, how do you want to say it? It ultimately gets down, does it release the islets or not? Mm-hmm. So I was wondering if you could comment on that, about how you view how you take this home to, how it applies to actually islet recovery versus other aspects.
Yeah. Well, I haven't thought about the islet isolation side of the matrix so much, because we've been, it's funny because it's kind of opposite concepts. Like, you're worried more about destroying the ECM, and I'm worried more about replenishing the ECM afterward. So I think it seems like you have to you have to successfully digest the ECM in order to get the islets out, and that's going to give you a better islet prep. And what we're working on is ways to support those islets once they're isolated. So I don't know if that was exactly what your question was.
Yeah.
It seems like there's not a good way around. You're not going to be able to leave the islet ECM intact. So what we're trying to do is to better understand the ECM so that we can incorporate elements of that back into the islet culture after isolation. And I think knowing that destroying or digesting a certain percentage of collagen is necessary in order to free the islets, then we just have to move forward knowing that that's a part of the islet isolation process. And so in order to support the islets afterward, we're going to have to restore some of that ECM that is lost.
Right. Dr. Swee from Georgetown asks, great work. Thank you for sharing the wonderful work with us. Could you point out among your assays, which one is the shortest turnaround time?
Yeah, sure. I mean, I think immunofluorescent staining is definitely the fastest of all of the methods. I haven't used RMS myself, so I think that's probably second. I know in our experience, the mass spec studies definitely take a long time. It takes a long time to run the samples. It takes an even longer time to analyze the analysis for those huge data sets. It takes a very, very long time. And then that's just to determine the relative intensities and identifications. Then we've got to analyze that data on top of that. So the mass spec definitely takes a long time. It's not a quick turnaround. But I think being able to run more samples simultaneously is making that go a little bit faster, and it's a little bit more high throughput now. And obviously there's a difference in the data you're getting out of that compared to just doing some immunofluorescent staining or something like that.
I had another question to follow up my first question. We try to make sense of how the enzymes actually free the islets, and one of the things that was real insightful to us was actually finding the structure function studies that were started in Japan and are now done with the people studying the 3D structure and mechanism of how collagenase works. And what it really gets down to in our mind is that you have to have collagenase because it kind of protects all of the proteins in the matrix. As you've shown in one of those slides with the histochemistry studies, the proteases, the matrix is very resistant to protease digestion, and it isn't freed up until which time you have the collagenase loosen that matrix up that you start getting decreases in protein staining. And actually, believe it or not, that one paper was very influential on why we proposed this concept. My question to you, and this again gets into the complexity and the reductionist, we think that really all you're doing is having to disrupt cell anchoring proteins. I mean, what holds the cells in? Mm-hmm. And do you have any ideas of what those proteins are? I mean, would you?
Well, I mean,
there are a whole host.
Yeah.
There's so many
and that's been studied a lot. It's not something that we have spent a lot of time looking at personally, because the endocrine cells express different integrins, and so that has been part of the approach to understanding which ECM components might be important to supporting islet health, is to also look at, well, which ECM proteins are they trying to bind to? They're making these different integrins and stuff. But also I think I don't know. I don't know a lot about how the integrin profiles of the islets differs from the acinar. So that could be a component of it.
Sure.
I think the islets express a lot of connexins and other cell adhesive proteins, and so that might also hold them together a little bit better than the acinar cells as well. But I'm not so sure about that.
Okay. I think your comment about the different species having different um, uh, uh, extracellular matrices is extremely relevant. And when we were doing the Liberase project back in the 90s, we found that each species had a different enzyme mixture. And I think one of the most intriguing findings that was done by the Groningen group and then later confirmed more recently within the last 5 years by the Japanese groups who are looking at monoclonal antibody collagenase, I should say recombinant collagenase enzymes, is that you can actually use just enzymes that are rich in class 2 collagenase to release rodent islets.
In rodents, yeah. Well, that's actually an interesting point too, because I think the fact that different enzyme blends are useful for different species suggests the importance of looking at the human pancreas specifically when we're looking at the matrisome. And we've had similar, so we do decellularization of the pancreas, and we've had some similar challenges where the protocols that are used in mouse pancreas, for example, are just not at all the same as the protocols we use with human or maybe other animals. And one of the big things that we found, I don't know if it has any relevance to islet isolation, but the lipid content of the pancreas is really, really different from donor to donor. And I think that's one of the big reasons that it's so much more difficult to decell the human pancreas compared to mouse. It's really full of fats. And so you have to get rid of those fats in order to in order to perfuse through the rest of the tissue. So that's one thing that we found. I don't know if that has relevance to digestion with enzymes either.
Right. I don't know. John Odorico said, excellent presentation in the Myers study. How did they quantify the percent digestion? Should the collagenase be targeting the collagenase at the islet interface versus acinar tissue only or some mixture? Usually the same question.
Yeah, so I should have a more clear answer. I think they were using a biochemical assay for measuring the collagen content of the pancreas in that paper. So they measured that before they digested, and then they measured after they digested. Now, I don't know, I guess thinking about what results after you digest the pancreas, I don't know what material they actually used, if they used the islets or if they used whatever was left over. I'm not sure. We'd have to look closer at the methodology to answer that question. But that's what they were measuring. They were measuring the collagen content before they treated with the enzymes and then after. And
you can see the question.
Yeah. Should the collagenase be targeting the collagens at the islet interface versus acinar tissue only or some mixture? Oh, okay. So I think that's asking the enzymes that we use to digest, do we want, we would want to break up the whole acinar tissue. We don't want to break up the islets themselves, but we've got to free the islets from their connections to the surrounding tissue. So I think it's got to target all of the ECM to some extent. Otherwise, you're just going to get lots of little bits of tissue, and that may or may not be what you're looking for. There's another.
Wait, Sui said another question at the end, and then Janice Kirchberg asked a question. Great work. Thank you for sharing the wonderful work with us. Could you point out okay, that was the older one. Sorry about that.
I think there's a question here about a predictor of human islet isolation.
Right.
Yeah, so I think that this is something we've done a little bit with. So when I was mentioning the lipid content, we did a study that we looked at the lipid content of the different donor pancreases that we get. And there was huge variation in lipid content of those pancreases. So we were interested in this because we just noticed this when we were decelling the pancreases, but we looked at islet outcomes as well. So we found that the pancreases that have higher lipid content also had higher lipid content in their islets, and that the islets tended to have poorer function. I mean, this isn't necessarily related to their isolation efficiency, but the one factor that we found that was common in those high lipid donors was a history of hypertension. So I think the reason I bring that up is just that there are a lot of different donor variables, parameters, measurements available that come with those different donor organs. And it might be worth looking more closely at those to correlate with the ECM content of the donors or to correlate with the islet isolation efficiency. I mean, there's already been correlations based on age, but I think other donor parameters may affect the ECM of the pancreas. I know just from looking at a lot of human pancreas tissue sections that it seems the ECM is pretty dramatically different from donor to donor. So I don't think there's necessarily an answer to that question right now, but I think it's something that would be important to look at, because sometimes we get a pancreas and I just look at the H&E of it and I'm like, oh, there's something very weird about this pancreas. But we would have never known that based on, we took that donor based on the information we had available, and nothing gave us a red flag that would be an abnormal donor. So
I just want to make a comment. We're having a discussion with Doug O'Gorman, who have done, I don't know, 1,500 isolations or whatever. And the point being is that you get an organ which will give you what you expect a certain outcome, and when it doesn't work, you just throw up your hands and say,
yeah. But
don't waste your time. No, but I mean, it's one of those things that's very difficult. I want to get to Janice before we sign off here. Had a question.
Yeah. So, I mean, we're working with for our mass spec studies, we're working with our collaborator Lingjun Lee's group, and they are really focused on novel mass spec technologies. So I think the strength of their method is that they've developed better techniques for solubilizing those ECM proteins and having better signal from those. We did try using decelled material, which is sort of like what the Orlando group did, but we found that we didn't need to run the decelled material side by side with the pancreas lysates in order to detect the ECM. So I think this is mainly just because mass spec technology has improved so much and is just continuing to improve so much, better solubilization of the ECM, and then the different methodologies that they use, which I can't really comment on right now, are enabling them to have better detection and better quantification of those proteins.
Okay. Well, thank you, Dan. We've run out of time, and really appreciate your contribution and educating us on the pancreatic ECM and islet isolation, how this all relates. So again, thank you for all attending. This will be on our website, so if you want to look at it later, you can view the video. Again, thank you, and I'll sign off.
Thank you.